keeping an eye on imaging. Discussion and commentary based on articles from JACC, Cardiovascular Imaging.
Welcome, ladies and gentlemen. My name is Stephan Achenbach. I'm here with Dr. Bradley Strauss, who authored a paper on microvessel formation in chronic total occlusions and the study by micro-CT, and with Dr. Renu Virmani, who wrote an editorial that accompanies that paper. Dr. Strauss, can you briefly tell us the setup of your study and what you investigated?
Well, we were very interested in looking at how microvessels formed in chronic total occlusions. So we developed a chronic total occlusion model in the rabbit femoral arteries by injecting thrombin into the femoral arteries to occlude it. And then we studied the arteries at specific periods of time after we developed the occlusion. And prior to removing the arteries, we injected them with a special material that allowed us then to visualize it on the micro-CT.
And I think the time points that you studied were 2 weeks after the occlusion and then 6 and 12 weeks. Is that correct?
And as well, we had an 18 to 24 week period as well, a very late period.
Now, you studied these arteries by micro-CT, and it seems almost unbelievable to me that you can see these tiny, tiny arteries by CT imaging. Can you briefly tell us how micro-CT works and what you have to do to be able to see this?
So it's an incredible technology that gives you resolution down to probably at least 17 micron size vessels, which are very, very small. What we do is prior to sacrificing, we inject through the aorta microfilm, which is a polymer that fills all the very small blood vessels down to the 17 micron level microvessels. And we allow it to sit for a period of time after we inject it for about an hour. Then we place the material into formalin to fix it, and then we place it in agar outside of the body. So we've removed it, and then it goes into a little box that actually has a CT scanner, very small device that can take pictures, and it takes about an hour and a half or so to completely scan the specimen. After we scan it, we then reconstruct the vessels using a software package where we can then outline exactly where they are.
So that's 1.5 hours of scanning for just one small volume, one artery, right, basically.
It's about 2 to 3 centimeter length occlusion in an artery that starts at 2 millimeter diameter and probably becomes much smaller actually in the center of it.
So what did you actually observe? What happens over the course of time after such an occlusion?
Well, it was very interesting for us because we managed to separate out the vessels that are inside the occluded lumen, which we call the intravascular ones, from the vessels that run outside the vessel, which we call the extravascular. And we could separate them out and looked at the pattern of when they formed. And we looked specifically at the proximal end of it, the distal end of the occlusion, as well as in the middle. The main finding of the study is that the extravascular vessels form first. They form very early on, and even 2 weeks after you have occluded a vessel, you can see quite an extensive extravascular pattern of these very small little vessels. And those remain still about at 6 weeks or high, but the intravascular small microvessels only seem to really develop around 6 weeks after. And they develop in a specific pattern. In other words, the vessels that are in the body of the CTO, not at the ends of it, seem to form separately from the ends, and later on they coalesce together. And it seems in many of the situations that there are probably connections between these extravascular microvessels and these intravascular ones within the body. However, at the proximal and the distal ends, it appears that these microvessels grow in from the actual lumen prior to and after the occlusion itself.
So, Dr. Virmani, how does this fit to the insight that we have from pathology about what happens in chronic total occlusions? Is this something entirely new, or was this expected to you?
There's some things new. For example, they're showing connections between the extravascular and intravascular. What we have observed, and we've done similar, not micro-CT, but we've done barium gelatin injection and seen how they connect to one another. And what we find in the human is it goes from the proximal to the distal end and covers the total occlusion part as a tortuous arteries. They could be single, they can be multiple as they saw. They also saw multiple, not as a single artery. And that these tend to be much more prominent at the ends, specifically the proximal ends are more circular, the lumen gets circular in the sense that it's concave, the lumen end, because it's usually near a branch point, whereas the distal end is more tapering, the occlusion. And then the vessels are very tortuous. We don't see a connection between the extravascular and the intravascular. I think there are two different phenomena. Now, it could be that their model was not an atherosclerotic model, was normal artery, and it may be totally different in case if they had an atherosclerotic vessel. So that's something that we need to learn. But it's still amazing that you can actually mimic this model. Now, the way they did it with thrombin to make a fibrin-rich and red cell-rich clot, whereas in the human, when we have total occlusion, we have an area which is very rich in platelets. You see, of course, you need fibrin, no doubt, to cause platelet aggregation, but it's predominantly platelet-rich. So it could be that these recanalized vessels may be quite different in the two models. But I think these need to be worked out, and I think there are good things about the model that, in a sense, they reproduced that you see connections from the proximal to the distal end. Whether they disappear, at what time they disappear in humans, unfortunately, we never have the history. Like they know when they occluded, So these are some guessworks we do, which they didn't have to do.
And a very brief opinion of yours, how do you think this information helps us clinically when we're thinking about CTO recanalization, for example? Is there any lesson that we can draw from this?
I think it tells you that if there is some recanalization, you want to go through those. This is, to me, is the most important thing. Am I right, Bradley?
The other point is we have a whole strategy of trying to grow new blood vessels into the chronic total occlusion to replenish the supply of these blood vessels which seem to disappear over time. So it may be a very important therapeutic strategy as well to restore these blood vessels at later time points.
One thing I will say, negative remodeling in the human is very extensive. As time goes on, that vessel gets very negatively remodeled. And I think that is true in the rabbit also, that negative remodeling.
Thank you very much for this interesting interview. Thank you very much, ladies and gentlemen, for your attention.